Welcome everybody to a new Global IMMUNOTALK. I'm delighted today to introduce you to my co-host, Lara Labartabajo, who is sitting in for Elena Zuniga. Lara is Dr. Lara Labartabajo, and you learn about her discoveries in Elena's talk. So thank you so much, Lara, for joining us. She just finished defending her thesis, and it's a real pleasure to have you co-host this talk with me. Thank you.
Thank you. It is my privilege for sure.
Okay, and so Lara will remind us briefly about the goals, and then I'll have the honor to introduce our speaker today, John Kappler.
Okay, well, thank you so much, Carla and John, for allowing me to co-host this amazing talk. So I just wanted to briefly remind everyone before we hear this amazing seminar. So the goals of Global IMMUNOTALKs are twofold. So essentially, this seminar series aims at benefiting and inspiring immunologists across the world in an egalitarian manner. So for that, talks are streamed online and are free access. Not only that, but they are always recorded and posted on YouTube on our channel, Global IMMUNOTALKs. Lastly, each of these seminars is followed by a virtual discussion and Q&A via Twitter with the hashtag GlobalImmuno that I will give instructions for in more specific ones at the end of the talk. Lastly, this seminar series also aims at increasing opportunities for scientific learning without the need of traveling. So this is an accommodating seminar series, and it's very environmentally friendly. And so we wanted to, before we go ahead with the seminar, we wanted to briefly thank all of the speakers that have so far kindly shared their science with all of us and taught us so many things. So this is just the updated calendar for the upcoming speakers. As you can see, followed by Dr. John Kappler today, we will have Dr. Russell Medzidov next week. So we look forward to having you all here again, enjoying these great science.
Thank you so much, Lara. Thanks. So, okay, so now I have the real pleasure to introduce our global immuno speaker today, Dr. John Kappler. John is a distinguished professor at the University of Colorado and at the National Jewish, where he has been a faculty member for 41 years, if I calculated that correctly. Now, one of the big honors, but also one of the big responsibilities of organizing the Global IMMUNOTALKs is to truly introduce giants like John. So we are delighted to have you, John, today. John initially trained as a biochemist at Lehigh University and Brandeis University. And this was back in the 60s. And I believe maybe he might tell us a little bit more, but this fundamental training in biochemistry shaped him as a budding scientist. Later, John moved to UCSD, where he did his postdoctoral work with Richard Dutton. And my understanding is that that's where he met also his lifetime collaborator, Dr. Pippa Marak. Now then, John moved to Rochester University, where he started his own laboratory and eventually moved to the National Jewish back in 1979, and that's where he has been ever since. Now, John is truly a giant in immunology. His work has really made fundamental discoveries on the properties of T cells and their MHC ligands. He has made discoveries on the nature of antigen processing and the presentation of the antigens restricted by MHCs, discoveries on T cell receptors, on positive and negative selection, and on T cell superantigens. As you can see, truly all textbook contributions. In more recent years, John has focused on the role of T cells in allergy and particularly in autoimmune diseases. And I think this is going to be a key focus of his talk today, which is entitled Neo-antigens generated by transpeptidation in type 1 diabetes. I believe he's going to discuss with us this idea that there's this unique generation of antigens in the pancreas, which is very different from what T cells see in the thymus, and that these can contribute to this lack of tolerance and eventual autoimmunity. Now, John has received multiple awards. Many of our speakers have, so we don't really go into details, but I do want to mention that he's an elected member of the National Academy of Science and the National Institute of Medicine of the United States. Now, on a more personal note, I have had the honor to visit John and his colleagues quite sometimes at National Jewish. I have always looked forward to the scientific discussions with you, John, and you have truly inspired me because of your sincere commitment and focus in science. So I'm very grateful that you have accepted to be a global Immuno speaker. Thank you for your efforts and for being here with us today.
Well, I'm very happy to be here. I want to thank you, Adelina, for the opportunity to participate in this great series. I hope that the fact that it's now on YouTube, that people will use it as a teaching tool for their students, and it will be more than just a one-shot deal.
Absolutely, and we have many people that then watch the talks in YouTube, so that has been truly inspirational and hopefully very useful for many. We get wonderful feedback. So before we start, we love to learn more about our speakers, and you've been in science for a long time. You have, experienced science in ways that some of our, younger mentees are, people that are joining us today have not yet. So we would love to know, what is the scientific discovery that was most influential to you when you started your career, and why? Can you tell us a little bit about that?
Yeah, I thought about this, of course, because you asked me before. Well, I started life to be an engineer. I went to an engineering high school and an engineering university. And while I was at university, I took a course in biochemistry from Professor Joe Merkel. He was inspirational. I had never actually taken a course that had any biology in it. I had taken math and physics and all that. And I just loved this course. It had a laboratory aspect to it in which you could actually design an experiment and do one and get the results and interpret them. And so I realized I didn't want to be an engineer. I wanted to be an experimental scientist. But you were asking for a turning point. Joe gave a whole course in biochemistry that I took. And one day he came in and didn't give his lecture, but he went to the board and he wrote up U, and then under it he wrote F. And the U was RNA, polyuracil, and the FFFS was polyphenylalanine. So Nuremberg and his colleagues at the NIH had just published the first deciphering of the genetic code. That they didn't know it was 3 at the time, but uracils encoded phenylalanine. I just thought about that and I said, the guys who did this, they went to the lab when they got that answer and they knew something that nobody else knew. And then they got to tell that to the world. And I said, that's the kind of life I want to live. I want to tackle problems that you have to design the experiment that will answer the question. And when you get the answer, it's kind of that eureka moment. You know something that nobody else knows, and you get to tell it to the world. So I would say that was probably a turning point for me. Joe went
inspirational
to me, and so there were a lot of other moments, but that just sticks out in my mind.
How inspirational! How important our professors are, right? And they can be so inspirational and encourage you to have the scientific life that you have had. Incredible. Thank you so much for sharing that. I'm absolutely, looking forward to your talk. So let's see if you can now share your screen and we can get started. Perfect. Thank you, John.
There we are. Okay. All right. So today I'm going to be talking about autoimmunity in general, but type 1 diabetes in particular. And when I became an immunologist now 50 years ago, the idea of I went to Dick Dutton's lab to study T-cell biology. That's what I wanted to do. And the whole idea of autoimmunity was a mystery. Why is it that the immune system goes after your own tissues? And why is T cells a big part of that, as it turned out? Well, in the next decades, we learned so much about how the immune system works, yet there were still questions that hung around, big questions. And you would think 50 years later we had solved all these questions, but we haven't. There are still questions that are not completely understood. The first one is that now that we know so much about control of the immune response, how is it that self-reactive T cells can break through all of the checks and balances that are put in their way that are supposed to prevent them from recognizing self-antigens? That operates in the thymus when they're developing, but it also acts in the periphery when they're being activated and when they have an effector function. So that's a big question. And the second question was, all right, autoimmunity starts. Why is it that only in a particular autoimmune disease, only certain organs are targeted, and why only certain antigens in that organ are targeted while others are ignored? So those are questions that have plagued us and I've been worried about for years and years. And that's why for the last 15 years, I've been working on autoimmune diseases, in particular type 1 diabetes, trying to get at least some information. I'm not going to answer these questions today, but I'm going to give you some idea of where it's going. So we're going to be talking about diabetes. So what is diabetes? I'm sure everybody tuned in knows that diabetes is an autoimmune disease that causes the progressive and specific T cell destruction of beta cells in the pancreas. And these are the cells that produce insulin, and obviously, and insulin is required to regulate glucose. And let's get a better pointer up here. Required to recognize, control blood glucose levels. And as the insulin production disappears, glucose levels become very high and they can't be regulated. And there's all sorts of pathologies. And of course, if you don't do something about it, death. So in humans who develop type 1 diabetes, they require daily glucose monitoring and insulin administration for life. So why is it that some people get diabetes and other people don't get type 1 diabetes? And we know a lot about that too. So first of all, let's talk about the progression of the disease. So here's a cartoon of the organ in which this all takes place, which is a small organelle in the pancreas called the islet of Langerhans, islets of Langerhans. In mice, there are hundreds to thousands of these. In humans, there's a million of these that are in the long arm of the pancreas scattered about. And these are these little organelles that contain the beta cells that make insulin. They have other cells as well. So we have alpha cells that make glucagon, the green cells here that make insulin and a bunch of other hormones, and the delta cells that make somatostatin. Most of these proteins are involved in either the health of the beta cell or in regulating glucose levels in the blood, up or down, as the case requires. So how does this immune system how does this response get started? It gets started in the pancreatic lymph node, the lymph node that drains this organ. And the beginning is antigens from or proteins from these islets find their way through they're well vascularized, they find their way to the pancreatic lymph node and they activate T cells who come back into the pancreas and they surround the islet, individual islets. They don't actually go in at the beginning and they hang around for a while. And then some signal, which is still not completely understood, signals them to go into the pancreas. And when they do that, they rapidly make cytokines and inflammatory things that destroy the beta cells in that particular islet. So the disease progresses as an individual islet specific event. Not all islets are destroyed at the same time, and this takes a long time to develop usually a long time to develop the disease, as islets are destroyed one at a time, all the beta cells in the islet, leaving this hawk that has alpha cells and delta cells, but no beta cells. So in the course of the disease, you might find an islet that has no beta cells. You might, in the pancreas, you might find another islet that looks perfectly healthy. And then you might find another islet that just has these T cells that haven't entered yet, but are collected around. This is called peri-encephalitis. So in a mouse, this takes a matter of months to go from this initial phase to getting diabetes. In humans, it can take years. Some individuals get type 1 diabetes as toddlers, and others, it takes until their 30s or 40s to show up, with the average age being sort of in the adolescent, young adolescent age. So what is it that determines that some individuals are going to get diabetes and other people don't get diabetes? Well, probably the most important risk factor are genetics. Studies in twins and studies in families say that about 50% of the risk of getting diabetes is genetic, and the other 50% is environmental or stochastic or unknown causes that generate the diabetes. So in the genetic compartment, by far, the thing that the alleles or the polymorphisms in genetic polymorphisms that give you the highest risk of getting diabetes are the major histocompatibility class 2 genes. In mice, the allele that is associated with diabetes is called IG7. This is present in the new mouse model of the disease. In humans, DQ8 and DQ2, these are MHC2 molecules in humans, 2 alleles. Those are the ones that are risk, and if you have both of them, you have the absolute highest risk of getting diabetes. There are other risks. These are polymorphisms in insulin, other genes, polymorphism genes that are involved in T cell function, T cell and activation, etc. So the MHC association with risk was discovered decades ago, and it predicted that CD4 T cells, which use class 2 molecules, or require class 2 molecules for the presentation of their antigens, they were going to be the initiating event, or they were going to be the a absolute seminal event in the initiation of the disease. And so we expected to find them specific for islet antigens, because they will go into the islet and destroy the islets. You would have thought that in all these years we'd have had this whole problem worked out, but we haven't. There's still years of research on this subject, and there's still questions about what are the nature of the pathological, or what they call the diabetogenic antigens in type 1 diabetes. So that's what we're going to talk about today. So why has it been so hard to figure out what these antigens are when it's a limited number of antigens? They're going to be present in the beta cells. Why has it been so hard to figure out exact epitope, exactly what the CD4 T cell is recognizing? And the answer comes from, or the problem comes from, the way in which epitopes or peptides that contain the epitopes are presented to CD4 T cells. So here is a typical MHC class 2 molecule. This would be the membrane down here on an antigen presenting cell where it's attached, and this would be the business end of the MHC molecule. And here's a peptide lying in the groove. This is an actual crystal structure of a MHC molecule with its peptide in the groove. So the question is, how does this peptide get in the groove, and what holds it there, and what determines which peptide a particular allele of MHC will bind and another allele may not bind? So there's two answers to this question. One is that there's a generic set of properties that are common to all MHC2 molecules, all MHC molecules and all peptides. And then there's a second set of properties that determine specificity of peptide binding. So the generic properties of MHC molecules that allow allow peptides to bind. I'm showing here, and this is again an actual crystal structure, where here's the peptide lying in a groove between the alpha chain and beta chain helices over here. And I've stripped away the side chains of the amino acids in the peptide and just showing you the interaction of the MHC with the backbone of the peptide. And all these green things are hydrogen bonds to the backbone of the peptide that are conserved in virtually all MHC peptide interactions. So you might say, well, if that's true, an MHC molecule is going to can bind any peptide, right? So we're going to see in a minute that isn't true. And also because the peptide can hang out of the ends, that is, the peptide is just not long enough to fill the groove, it also can hang out of the ends. A peptide might bind in different positions in the class 2 molecule. Furthermore, these positions it can bind in are not continuous, they're digital. That is, every time if this peptide wanted to shift into another position, it would have to go exactly an amino acid at a time because it would have to reestablish these backbone interactions. So they're very important in limiting the way the peptide binds, but also the position that the peptide binds. So if this shifted over one to the next position, it would have to bring these nitrogens in blue or this oxygen from the next amino acid into this position to make this interaction. So where does the specificity come from if it's just a generic capture of the peptide in the groove? The specificity comes from the side chains of the peptide and the allelic differences between one MHC molecule and another. So I've taken this one of these structures up here and tilted it up on end and stripped away the beta chain, so you can look at the peptide packed up against the side of the alpha chain. And what you can see is there are 4 amino acids pointing down and a bunch of amino acids pointing up. These are the amino acids that are going to interact with the T cell, and these are the amino acids that are going to stick down into the groove and determine the specificity of binding. And you can see in this case, we have a lysine here, we have a glutamine here, and we have two aliphatic amino acids, isoleucine and leucine. In this particular MHC molecule, at the bottom of this groove is an acidic amino acid to interact with this basic amino acid. So this pocket that's binding this one absolutely prefers to have a basic amino acid in it. This amino acid here makes a big hydrogen binding network with the bottom of the groove, and polymorphic amino acids in this region capture the end of this side chain in a hydrogen bonded network. Over here, we have hydrophobic pockets that these 2 amino acids bind in. So you can see that although theoretically every peptide can bind in any position in the groove, it's limited by this interaction here. And this is going to become important when we're talking about type 1 diabetes. What are the nature of these amino acids pointing down into the groove of the MHC2 molecules that are important in diabetes? Okay. So we're going to go a little short history of the two of the major epitopes for CD4 T cells in type 1 diabetes. I'm going to emphasize the work that's been done in the NOD mouse, but particularly in this first little history for insulin, many of these principles have been confirmed in humans as well. So the first protein we're going to talk about is insulin itself. It's been known for years that insulin is one of the autoantigens in type 1 diabetes. You can tell that T cells are involved in this response from the beginning because you can detect anti-insulin antibodies very often in patients who are in the pre-diabetic stage. So T cells are being activated because you don't get the antibodies unless T cells are helping the B cells to do it. So the first major paper on insulin came from George Eisenbarth's lab, where Dale Wakeman used a culture system that Katie Haskins' lab had developed and found a whole bunch of T cell clones, CD4 T cell clones, that reacted to this peptide from the beta chain of insulin. So you would have thought, now we know what the peptide is, you know, apply those rules I was just talking about, we'll figure out how that peptide is bound to the molecule in the NOD mouse, and that will tell us what the antigen is. Well, in fact, instead, we've had 25 years of arguments and controversy about what is this epitope in this peptide, or epitopes in this peptide. How do they bind? And remember, I was talking about those registers. That is, you can digitally shift this peptide. It's a 15 amino acid peptide. It has 7 theoretical positions it could be in the IgG7. Which ones of those are the really important ones? So lots and lots of arguments, and I would say that the first real extensive look at this subject occurred when Mateo Levisetti was doing experiments in Amelia Nanaway's lab and published this paper in 2007. They concluded from their experiments that the peptide was bound in 2 different ways, all right, 2 different registers. We're calling them register 1 and register 2 here. And that some T cells like this register and some T cells like this register, and they called them A and B, different types of T cells. And you can see that vis-a-vis what I was telling you about what has to happen for a peptide to shift, it isn't just being translated sideways, it has to roll over to bring new amino acids into the pockets and put new amino acids on the surface. So this epitope is as different from this epitope as 2 completely different protein epitopes are from each other. So they did a whole bunch of experiments and found a bunch of T cells that they say responded to this and this. And they used some tricks like truncating the peptide from either end until the stimulation went away for the T cells, and some mutational work, and they came here. So George Eisenbarth asked us to look at Dale Wegman's sequence clones and see if we can't find out how those clones are binding to IgG7. Or is it this way, or is it some other way? So a graduate student came to my lab and determined to work on type 1 diabetes, Brian Stadinsky, and he started using the tricks of the lab to look at George's clones to see how did they respond. And he looked at a number of different registers, but I'm going to put 3 up. So he's looking at the 2 registers that the Nanaway lab had said, and he used a 3rd one as a sort of a control. And he used the tricks in our lab, which was to mess around with anchor residues. These red ones are all the anchor residues. They're the ones that stick down into the pocket and determine specificity. So he made them more optimal or less optimal. And we also engineered disulfide bonds between the peptide and the MHC, so that when they bound in a particular register, they could not move. They had to stay in that register. So this allowed us to make homogeneous MHC peptide complexes that were only bound in one register. So he looked at George's clones this way, and he concluded that all George's clones were bound in this register, not the registers from the Nanaway lab. So I was very suspicious of that. Said, you know, we better really make sure we're right, because you know, just don't want to publish something that's wrong. So we gathered a bunch of clones from the Barbara Davis Center and from the Nanaway lab, and we used these tricks of fixing the registers. And what Brian found was that all of the clones, whether they were A or B, recognized the peptide bound in this register. So that was, of course, a very controversial finding. So how did we do it? What was it that now makes us believe that this is the way the peptide is bound? So, and not this way. So let's take a look at the different peptides that Brian had. So if he has the unadulterated peptide, the wild type peptide, it's a very weak antigen for both type A and type B T cells. And so we looked at, and we said right away, it said, well, if we wanted to bind in register 3, this is a terrible anchor residue in IgG7 for that class 2 molecule. So he mutated that to E. And lo and behold, the T cells continued to respond. So now the peptide is found in register 3, but now it has a perfect anchor residue here. And the type A T cells all responded about 100 to 1,000 times better. Later on, when Brian left, so we're looking back up here and we say, oh, look, what Emil was doing, or his lab was doing, they were truncating this peptide. And when they removed this R, they didn't lose the response, even though down here that would leave the pocket a little empty. What we did is we took away this inhibitory amino acid, and that's why they mistakenly thought that it was a register 2 ROP3. So we said, well, what about up here? Maybe the same thing is going up here. These are bad amino acids here. So we mutated this amino acid as well to take the side chain off of the surface, and so that the T cell wouldn't see it. And then we got an astonishing result. That is, not only did they continue to respond to this peptide, but now the specificity flipped. Now all the type B cells were happy, and the type A cells still responded, but weakly. So let's look at this data to what happened here. So here's some data from Fran's paper, and then we've done many, many experiments along the way. I'll give you some additional data from Yang Wan's paper. So we're going to look at one of the prototypical type A T cells. This is from the Barbara Davis Center, and watch its response. So here is this very weak response it has to the wild type peptide. Have to add a lot. So we're titrating in the peptide and measuring the level of the T cell response. If we just make this one mutation here, this gets 100 to 1,000 times better in stimulation. So that's exactly what we predicted would happen if it were found in register 3. If we made this additional mutation here, we lost this increase in activity. So this said, not only did we have to make the peptide bind better, we had to leave this E here for that T cell. So what about the case of the type B T cells? Again, if we had the wild type peptide, very poor response. But now, if we made this mutation, it didn't get any better. But if we made this mutation, we had this flip around response. So we were convinced at this point that we had solved the problem. There was something wrong with the natural peptide that required you to mess around with the C-terminus, put it in a different amino acid there, or in Emil's case, truncate it in order to get it to be recognized strongly. And we went on and did lots and lots of experiments with these. But eventually, to solve the, put the nail in the coffin here, we did the crystallography. I want to say before that we collaborated with the Barbara Davis Center and with the Ben-Aurora Institute to look at human T cells, and we found that the human T cells that reacted to this peptide have this phenotype here. All right, so let's look at the crystallography. So I'm not going to show you the whole story of the crystallography. I'll just show you the important points for these two amino acids. So this was done by Yang Wan and Xiaodong Dai's lab, who has been a collaborator for 20 years and doing crystallography with our lab. And here was the crystallography of a type A T cell. And this is a piece of that T cell. And here's the peptide that we mutated here, sitting in the groove. And you can see that P9 position now has got this beautiful acidic amino acid that it loves. And here's the P8 amino acid, the natural one, sticking straight up out of the groove and interacting with the TCR. If we take this away, we break this interaction, and we lose a lot of the response. We have a reciprocal story for the type Bs, that here's the type B peptide sitting in the groove. Again, at the P9 position, we have this nice acidic amino acid replacing the R, right, over here. But now we've taken the side chain off of this amino acid and by substituting a glycine. And now the part of the TCR that wants to interact with this has settled down right on top of the backbone of the peptide. And if you put this back in, you push this TCR off of it, and it accounts for why the response is so poor with the wild type amino acid here. So keep this in mind now. We're going to go look at some other data now, that for insulin, we have to do something to the end, the C-terminus of the peptide to get it to work. All right, so the conclusion so far is that B923 peptide is presented in register 3, but it's a lousy antigen in register 3, and it has to be mutated or modified in some way to make it bind better to IgG7 and make it interact well with the TCR in some cases. Okay, so now we're going to take on another antigen. This is chromogranin A. So chromogranin A was another antigen that was discovered early on to be involved. It's another protein from the beta cell. And the initial experiments were done in the Haskins lab, and they isolated a couple of clones. They're called BDC for Barbara Davis Center, because Katie was there at that point, 2.5 and 10.1. Now they were discovered to be very strongly reactive to islets and very diabetogenic, that is transferred into another animal, they cause diabetes very quickly. But they didn't know the antigen. So this is 1989 when they were working on this. And people worked on this, and they didn't get anywhere. And finally, some years later, 2 studies, one a collaboration with the Haskins lab from Japan and another one from La Jolla, they bit the bullet and did some library studies and this a technique called positional scanning, in which you sort of mutate amino acids one at a time along the whole possible peptide and see if you can't come up with something that stimulates. They both found related peptides that strongly stimulated either just BDC 2.5 or both BDC 2.5 and 10.1. This is just one example and this group found many peptides. But the thing they had in common was this motif at their C-terminus, right? They had tryptophan, and then the R, M, and then the acidic amino acid. Remember the famous acidic amino acid that's needed for the P9 position in IgG7. But they had quite a lot of variation here. So again, Brian Sadinsky, when he was in our lab, we got in a collaboration with the Haskins lab, and he used our library system, which was a display system in which we made MHC molecules that had a whole library of different peptides in them genetically and expressed them on the surface of an insect cell and screened. And he came up with another peptide, and he came up with this peptide. So at one level, he was disappointed because he came up with the same thing everybody else came up with, but it was subtly different at the big front end. And when people had screened libraries of proteins and everything, looking for something that would be like this, they didn't find anything. But when Brian looked at libraries, which were a lot better now, 10 years later, when he looked at libraries for this, he found right away that chromogranin had a sequence in it that resembled this. So here's the sequence. The sequence is here, right in the middle of the chromogranin gene, and guess what? It had that same motif at one end, but it was very different from these other other peptides at this end. So undaunted, we got together with Katie's lab, and her lab and our lab tested this peptide and found out it had no activity at all. That's a big disappointment. So licking our wounds, Brian and I went back to think about this. Why did this fail? And then, of course, now at this time, we knew a whole lot about how peptides bind to IgG7. Remember all those rules about what's going to fit in this pocket and what's going to fit in this pocket. And we knew that this R here was exactly the wrong amino acid for this 4 position down in the groove. And Brian had done some mutagenesis work that showed that if you take in his protein, peptide, and you put a K there, it just kills the peptide. So he's remembering back to the Nanoway experiments and our own experiments, we said, maybe what's going on here is these amino acids, especially these 2, are inhibitory for this peptide being recognized. And at the same time, we realized that those 2 amino acids were a processing site for the convertase proteases that take the larger protein, chromogranin A, and make smaller proteins out of it. And one of those proteins is a peptide called WE14. So it's cleaved here and cleaved at another double positive charge here to generate this peptide. Very well documented in the literature and very conserved in evolution, this peptide, although its function is still not known. So we said, okay, it's a long shot because we're going to end up leaving all these positions empty. Let's test this peptide. And so along with the Haskins lab, we tested this peptide and it was active. So removing the inhibitory amino acids allowed this specificity to come through. It's a very weak response, but nevertheless, it was real. And so the chromogranin knockout mouse was obtained. We got that from Sushil Mahata in San Diego. And lo and behold, that animal lacked the antigen for these T cells. So the paper was published, and the paper was published, this paper here, and we decided that chromogranin was required for this antigen. But, it was very unsatisfying that this was such a weak response. And Katie's lab had already partially purified the antigen from islets, and it had a higher specific activity. So we were convinced that something had to be going on here to make this better. And of course, we remembered our insulin experiments in which it wasn't enough to remove the inhibitory amino acids. We had to add back good amino acids at those positions to complete the epitope. So what Brian did was he used these 4 amino acids from the library peptide he found, and he fused it to the end of WE14. And lo and behold, we got enormous increase in activity. And so this was published in several places since then. So let's look a little data again. So here is the data that nailed this, this was published in a paper by Jen and Wang in PNAS, in which we showed that when we made this addition to the N-terminus of WE14, compared it to WE14, we got somewhere between 100,000 and 1 million-fold increase in activity. So this convinced us that there's something going on that at the N-terminus of WE14 where we have to add something here, ideal amino acids, in order to get a response. So just to nail this down, again, Yongwan and Xiaodong Dai's lab solved the structure of the BDC 2.5 recently, and also he solved the IgG7 complex as well. But here I'll just show you one picture from those structures. Here is this RLGL attached to the end of WE14 as it sits in the groove of IgG7. And again, you see what by doing this, we put back 2 anchor residues. We put a P4, we put the ideal L, and at this position, we put the ideal R for position 1. And this leucine is sticking up, interacting with the TCR, and would be a multiple van der Waals interactions. So now we had 2 antigens that we have 2 antigens. So just to summarize that, the natural epitope that would be the natural protein from chromogranin A is inactive for these CD4 T cells unless something is done with the 4 amino acids at the end of that epitope. You can eliminate them and you have a weak antigen, or better, you can replace them with good amino acids and you get a super agonist peptide for the diabetogenic T cells. So, the major epitopes for both insulin and chromogranin require some kind of insulin amino acid replacement within the parts that are going to interact with IgG7 in order to be effective. You know, that's all well and good, but you know, you can make those peptides, but are they actually really made in vivo? And how? So is there a mechanism for replacing amino acids in proteins in vivo? So on a visit to Yale, giving this data and scratching my head about it, Pete Cresswell pointed out to me that there's a literature that I should have already read but hadn't on transpeptidation, and that people had been working with this in the early 2000s and had shown that transpeptidation can create antigens, peptide antigens for CD8 T cells. So we immediately saw this as the answer, maybe perhaps, to our question of where did these changed epitopes come from? So what's transpeptidation? Transpeptidation is excuse me. Transpeptidation is a reverse protease activity. It starts when one of these proteases that can do it cleaves a protein and stays connected to its N terminus, and then eventually water comes in, breaks the bond, and you end up with a complete proteolysis. But if there is a peptide with a free N terminus, it can get in there first and create these hybrid peptides. So there's a whole literature that goes back in the 1930s on transpeptidation, and there's a bunch of papers now that say that the threonine proteases, which have an OH instead of an SH here, can do this in the proteasome. So we wanted to know whether there were transpeptidation reactions that could make our chimeric peptides. And we knew from the literature that the secretory granules that had all these proteins turn over in beta cells by fusion with lysosomes. And so they are exposed to all kinds of proteases, particularly cathepsins, during that process. And this would be an ideal place, just like the proteasome, for this reaction to take place. So we decided we were going to make an effort at that. So we took a look at the beta chain that we were working with, and we had two places where we had to cleave to get rid of the stuff we didn't want. And then we had to find donors that would come in and go with that. And so we looked at the Cpeptide, which is just downstream of the beta chain in insulin. And there were just loads of places where you could have, if you cleave this peptide somewhere and create a new N-termini here on these peptides, they could either be donors for type B or type A T cells. So we made a whole bunch of these guys and tested them, and they're all active. They're all extremely well active. So you can recapitulate our mutagenesis data by fusing pieces of C-peptide to the right place in this beta chain peptide. Unfortunately, we have not yet found a protease that will cleave at this place, so we haven't been able to do the transpeptidation reaction there. So I'm going to spend the rest of the time talking about the W14 and what kind of acceptors might be helpful for that. So we already have the RLGL that we had from the, but are there natural sequences in beta cell proteins that could do this? So we scanned these proteins looking for potential acceptors, and we ended up with a list of about 20, and these 8 turned out to be highly active. So one from Cpeptide, some from secretin 2, some from 3, and importantly, some from chromogranin, because this would be an internal transpeptidation. So we were very interested in that. So while we were doing all these experiments, Tom DeLong had started doing these kinds of experiments as well. He had fused a piece of Cpeptide to the end of W14 and created an epitope, and then went on to actually find it in the pancreas, that peptide. So we said, okay, for our transpeptidation reaction, let's start with that one to see if we can make this peptide by a transpeptidation reaction. So the first thing we had to do was look at Cpeptide, here's where this piece we want is, and see what's it going to take to get a cleavage here with one of these enzymes that make this covalent intermediate. We tried a bunch of enzymes, and the only one that worked of the ones we tried was cathepsin-L. So when we cleaved this with cathepsin-L and did a mass spec analysis of it, we found that lots of fragments that the cathepsin-L had cleaved right at the exact point that we were interested in. So we repeated that experiment in a transpeptidation reaction. We put in the Cpeptide as the acceptor with the buried piece that we want in the sequence. And then we put in a lot of W14 as a donor and did the same reaction again, and then looked to see if we had any activity in the digest. So here is a synthetic version of that peptide, just as a positive control. It's very stimulatory, just like the other ones were. And the two peptides that are going to take part in the reaction have no activity, but when we treated them, this combination with cathepsin-L, either from recombinant cathepsin-L or liver cathepsin-L, we had great activity. So that was one of those eureka days when this experiment worked. So now we had to prove that what was happening here was, in fact, we were getting the peptide we were talking about. So we ended up doing a reaction with smaller versions of the acceptor and donor. Here's a piece of Cpeptide and a piece of W14. So the mass spec would be easier in the analysis. And we did the digest. We tested the digest on these two hybrid 2 T cells. We analyzed the digest by mass spec, screening for the peptide that we were looking for. And we did a comparison of the peptide when we found it to the synthetic peptide to make sure that the mass spec analysis was telling us the right thing. And then we made synthetic version of the peptide we found and showed it was active. So I'm going to show you that for the insulin Cpeptide acceptor. So here's what went in the reaction. Here's the enzymes. And again, we got, for these two hybridomas, we got great activity for both of them. When we did the MS analysis, we found, and this is the predicted fusion that we were going to see, the chimeric product. We found the chimeric product, with both enzymes, both enzymes, we found that peptide. So when we did the mass spec analysis, the double mass spec analysis, and looking at the fragmentation pattern of the peptide during the second phase of the mass spec, we got this pattern of what they call Y and B ions, truncations from the one end of the peptide or the other end of the peptide. And when we put in the synthetic version of this peptide, we got exactly the same pattern. So this told us that we had found we had identified the peptide properly. And then when we synthesized the peptide, it stimulated both hybridomas. And here's a control T cell down here that it didn't stimulate. So that was great. We had what we were looking for. We had a reaction and an enzyme that would generate this peptide, which was known to be present in the beta cells of diabetic mice. So let's come back to this list. We also tried to generate this peptide with other ones, because we had tested all these peptides and shown they were highly active. Could we make any of those? And it turned out we could make 4 of them in this cathepsin-L experiment. So here are the digest and the activity of the digest. tested with both BDC 2.5 and BDC 10.5. We had activity with one or both of those T cells. So we went on and looked for the peptide in the soup of these, in the digestion. And in every case, we found the predicted chimeric peptide. And when we did the mass spec and we did the analysis, the synthetic version of this all matched very well. So we had very, very high confidence that we had identified the peptide. And then, of course, when we synthesized the peptide and tested it, we also had absolutely the predicted activity. That is, sometimes BDC 2.5 was stimulated better, sometimes BDC 10.1 was a little bit stimulated better, and sometimes both were stimulated the same. So, final conclusions. The natural epitopes for diabetic T cells, specific for W14 or that have been identified over the years, are likely to be, the actual epitopes are likely to be chimeric peptides that are formed by this chronophagy fusion of lysosomes with the secretory vessels in the beta cells. And this is by a cysteine or serine protease involved in transpeptidation. I think pretty confident that this is, you know, the Cox postulates just about have been totally confirmed here. The peptide is there, the peptide can be formed by this reaction. What we'd like to do now, and we got interrupted in doing these experiments because of the COVID mess, we want to go back to our insulin story and find a protease that will cut at the right place to test whether or not these segments of C-peptide can do the same thing. And likely they will work if we can find the right enzyme protease to do that. So, transpeptidation during chronophagy, during the fusion of secretory granules with lysosomes, it's likely that it also can play a role in other autoimmune diseases of neuroendocrine tissues. So, you know, this could be the adrenal glands, this could be nervous tissue, this could be narcolepsy and the hypocretin issues of a protein. Now we have this mechanism in mind. We should be going back and looking in those autoimmune diseases to see whether there isn't a role for transpeptidation in during the turnover of the secretory granules in those tissues. And finally, I think the most profound implications of this reaction being so common in vivo and in any proteolytic reaction involving these antigens. This is a very common side reaction to get these chimeric peptides. We should be going back and looking at databases, because all the databases that are out there up until recently have stripped peptides from class 1 or class 2 and looked at their sequence, trying to match them up against the natural sequences found in proteins encoded in the genome. It's only been recently that some algorithms are being developed to be able to screen those elutions from class 1 and class 2 to see if they can identify the chimeric peptides. So Mishta's lab has been working on this. There's some other labs that have been working on this and have developed pretty good algorithms now for finding these peptides from class 1. It's a much harder problem in class 2 because the peptides can be way variable in length, and that makes the computational end of that search much more difficult. But it's coming. Someone will figure out how to do this in the next year or two. So that's where I'm going to stop. I'm going to thank again everybody for listening. If you made it to the end, through all that detail. And thanks, Carla and Elena, for inviting me to give this talk. Thank you.
Thank you so much, John. That was fascinating. I really appreciate your long-standing efforts in trying to solve this. I personally very much enjoyed the clarity of the results. And
I forgot a slide on that. I really have to do this.
Okay, yeah, you should go. Okay, please,
I
go ahead.
Left there, right?
Yes, of course. We absolutely have time.
Got so excited about the results. These are all the people over the last 17 years who've been working on this project and did all the heavy lifting, okay? So in our lab at National Jewish, of course, Pippa, who has been my partner in life and my partner in science for almost 50 years now.
Wow.
But I want to really give a special heads up to Brian Stadinsky, who dragged us into this field. We were working on other aspects of autoimmunity, and he said, I really want to do this. And so, and it's now become one of the major projects of the lab. He did the seminal experiments that got us started. And I also want to give a special shout out to Fran Crawford, who was with us for 20 years. She's now retired, drinking a gin and tonic somewhere with a view of the Rockies. And Fran has been our go-to person for biochemistry and protein expression, etc., for 20 years. And thank you, Fran. And then there are other people who have been in the lab, especially Janice White and Neo Jin, who have contributed to this. And in fact, Neo has her own story to tell. That's a different story from this one about type 1 diabetes that we're writing up now. Harsha Kirby was a rotating student through the lab who did a critical experiment a long time ago. So most recently, I want to shout out Brendan Reed, who's been the person in charge of the transpeptidation experiments. He's worked with Fran on these experiments now for the last several years, and also with the mass spec facility at the university. Then, of course, Sheldon Dye, who's been my collaborator in crystallography for 20 years, and all of the structures on type 1 diabetes that we've done have come out of his lab, most of them done by Yang Wang, who did a fantastic job of solving these structures. And then in the beginning of the chromogranin story, we collaborated with Katie Hassan's lab. So now the second people are at the Barbara Davis. I want to point out George Eisenbarth, who encouraged me and wanted me very much to get involved in type 1 diabetes, and then a whole crew of people there who have over the years have given us cell lines and constructions and ideas and done experiments for us, especially when we decided to look for this in human cells. So I can't go through what every single person did, but every one of these people have contributed intellectually and scientifically to this story. Kurt Hansen's mass spec facility has been absolutely essential for us getting the answers that we have. And I want to really point out Ryan Hill, who has stuck with us through the whole COVID experience. And finally, we got all the data we needed to have this paper submitted. At WashU, Amelia Nanaway and Lavisetti gave us clones and T-cell clones to study. At the Ben Aurora, likewise, we got human clones from Bill Kwok and Jean-Bao Yang. And at San Diego, Sushil Mahadis gave us the knockout mice. So, and here's our funding, NIH, HHMI, lots of people have contributed money to this project. Okay, so now I'm done.
Okay, but thank you so much. Thanks to you and to all your colleagues for this terrific work. I was truly blown away by the precision, right, of biology. It was really a delight. So we will now say goodbye, but before saying goodbye, if I have Lara, let's see there, I would like to remind people that they can ask questions via Twitter. I have already put a tweet, and John is going to use our Immuno speaker account. So thank you, Lara. That's great. And so if we go to the last slide, we are now seeing, thank you, Lara.
You're seeing.
Yeah, we're saying thank you to the speakers. If you go to the next slide. And I think now our audience is very, to the next one, let's see. But our audience really knows nowadays what we should do. The last slide will remind us that there's a tweet that I already included in Twitter that says, ask questions for Dr. John Kappler here. And just remember to put the hashtag global immuno, and John will be answering the questions via the Immuno speaker account. So I just said it, and this is all fine. John, thank you again. And Lara, thank you so much for co-hosting this with me. We will be answering, well, John will be answering the questions through Twitter, and we, of course, will be putting this in YouTube so people can enjoy John's talk again or for the first time. Thank you, John.
Okay, thank you.
We'll be in touch.
Yeah.
Thank you. Bye-bye.
Bye.